chair for the committee, and he is going to talk to us about CMEC and the translational immunology and the DART study.
Thank you. Thank you, Dr. Kirchhoff. Can see the screen. Can you all see the screen? All right, so I'll start. So I will give you an update on translational medicine portion of the DART trial. We forged collaboration between CMAC and us, SWOG. These are my disclosures. So we're going to talk about the objectives, the elements of TM, and the recent evidence behind the hypothesis that we have, and talk about the academic and commercial collaboration that we have, focusing on CMAC. So we want to replicate what we saw in other histologies such as renal and melanoma, utilizing the nivo-ipi, which at that time when we first ran trials did not have much correlation with other biomarkers. So you want it to be histology agnostic trial, but then really use this as a chance to learn about the rare tumor. So not only about the biomarkers of response or resistance, but really understand the genomic, transcriptomic, and proteomic and immune landscape of rare tumors. So we're going to talk about genomics, transcriptomics, proteomic, immune landscape in 4 parts. So this is how it all happened. It started as a NCI match precision oncology medicine that was focusing on rare tumors, and that's when we proposed DART trials. And this was the time when there were a lot of screen failures from next generation sequencing assay. So whoever were unmatched or progressed and didn't have another match treatment arm, then they would fall into DART. So we initially had access to the NCI match TM data, which is listed here. And now we have a separate SWOG repository because we were separated later on when the NCI match trial reached full capacity. So the first section is the genomic traits. So we are focusing on PM tumor genomic traits such as tumor mutational burden or DNA damage response gene aberration, or in other words, genomic instability. So I think it's a great platform because we have over different, like over 50 different histology cohorts in one trial. It's a true basket trial, and we're able to test what we found, whether what we found in immunogenic tumors can be replicated in other rare tumor types. This is a very interesting graph because this is not looking at what a certain clinical trial by trial. It's just a understanding of TMB landscape in different tumor types and the object response rates reported in immunotherapy trials in those histologies. So it's very crude cross-sectional cut of the landscape. So we thought this would be a nice representation of our hypothesis, where is there really a pan tumor marker, genomic marker such as TMB that will be associated with response. And looking at multiple immunotherapy clinical trials in different histologies, Sloan Kettering Group have identified some trend that yes, overall survival can be predicted according to TMB level. But this may be a phenomenon that is mainly driven by a certain type of tumor, and I'll get back to that later. And this is the non-small cell lung cancer experience using immunotherapy. And you can see here, if you use first-line nivolumab, which this trial really didn't pan out, CheckMate 06, if you look at the right side graph, the overall survival really did not change according to tumor mutational burden. And then this is the lung cancer trial that used atezo, single agent, everyone gets it upfront, And the response rate did change according to TMB, with the caveat this was the blood TMB that they used. But then you look at the overall survival, no really big difference in overall survival according to tumor mutation burden. So is it just the effect of the early response rate with no obvious beneficial effect on overall survival? We really don't know. Maybe it depends on histology. And this is bladder cancer experience where really TMB high and low, you can see difference in response as well as overall survival. So what we didn't see in lung cancer was actually seen in bladder cancer. This is also the same atezolizumab given as a single agent. So this is nivo-ipi early experience in non-small cell lung cancer where the benefit were found regardless of the PD-L1 high or low, or present or negative. And this is the pivotal trial, Checkmate 227, that led to nivo-ipi approval in lung cancer. And there's initial reports showing that it has major impact in high tumor mutational burden. But the later papers showed that the effect was really also seen in both TMB positive and negative, and PD-L1 positive or negative didn't have much impact in overall survival. And this is a subgroup who benefited from this nivo-ipi approach. And as you can see, TMB high-low, PD-L1 high-low really didn't make any difference in terms of overall survival outcome in this group of patients who received nivo-ipi as a frontline regimen. So TMB, as of now, remains as a companion biomarker for pembrolizumab study across all different histologies. 10 is their cutoff, and this is really based on the Keynote 158 overall response rate being about 29%, and these are the histologies that you can see in that trial, which is kind of like DART. And this is by TMB, the response rate difference. But again, understanding that there is a collection of different histologies, there was no overall survival difference. And this also taught us, as with many other trials, that tissue TMB and PD-L1 are really not correlating with each other. They're independent of each other. So this is a very interesting article that just came out last month. High tumor mutation burden fails to predict immune checkpoint blockade response across all cancer types, coming from Timothy Chang group. And what they noticed was that I guess the reasonable expectation is if you have high neoantigen load in the tissue, then you would see more CD8 T cell cytotoxic effector T cells present in the tissue. And endometrial cancer, bladder cancer, you can see on the upper graph, there's a nice correlation. But if you go to prostate cancer, breast cancer, you really don't see that kind of correlation. So only in what they defined as a category 1 tumor type, where there is that tight correlation, is where we see the impact of tumor mutation burden high as predictor of immunotherapy. But in other groups where they categorize as type 2, they really didn't see that relationship. And this is in different tumor types. As you can see, low TMB and high TMB, the response rates really did not change in separate clinical trials. So this is really different than the NEJM paper, nice slope that I showed you. That was a cross-sectional graph showing a histology as a aggregate, but this is just individual clinical trial. And that in that trial, if you separate them by low high, do you see correlation with response and the differential TMB values? If you look at the metastatic breast cancer, glioblastoma in the lower graph, you actually see detrimental effects of immunotherapy if you give immunotherapy in TMB high group, which is kind of surprising. So they did put these category 1 and category 2 as an aggregate, yes, in category 1, you see some benefit. Category 2, you don't see benefit. And for overall survival outcome, it may be even detrimental. So stay tuned, and we'll look at rare tumors to see if any of these patterns pan out, and we will see whether what their conclusion can really be applied into the DART trial. So many interesting things are still coming out, so we are up to date on current science. And we're using immunotherapy only combination, and the studies show that the TMB really is not predictive at all when you combine it with chemotherapy, and which is confirmed with Keynote 21 and 189. And here, histology really matters because small cell, yes, TMB, it seems to have impact on overall survival with when you give nivo/ipi. Now turning into different pathways. You can have a high TMB, but it might not have any impact if you have the immune effector pathway blocked by either JAK1/2 gene mutation, loss of function mutation, or beta microglobin gene required for neoantigen sensitization, or HLA class 1 genotype is defective, then even with MSI high colorectal, you're never going to get response. And this was what was published in NEJM and also by the Tony Rivas group. And then we know that chromatin remodeling genes such as PBRM1 and many others can play a role in immunotherapy sensitivity. And also, not all neoantigens are created equal. We believe frameshift indel mutations are very different than others. And that is because the frameshift will create more change down the road in amino acid, leading to a differential protein folding, making probably neoantigen more sensitized to a TCR. So this is our group's observation, where if you have frameshift indel present versus not, just that simple dichotomy will be able to predict response and survival outcome from non-small cell lung cancer patients getting immunotherapy. And then the TMB landscape usually does not always correlate with the FS indel landscape. This is looking at all foundation medicine patients that are sequenced. And then another genomic trait that of significance that Dr. Kurdrak, Dr. Goodman had reported is the PDL1 amplification. Sort of extrapolating from what we knew from Hodgkin's lymphoma, 0.7% incidence across various tumor types. We have created a separate arm within DART. This is the only arm that is not using nivo/ipi but single nivo, and this is the JAMA Oncology paper reported by Dr. Kurdrak's group, and response rate of 66% with PFS of 15 months. We are struggling to accrue appropriate patients for this trial, although we've kind of made it very clear. I think we've had sites enroll PDL1 high expression by IHC being enrolled into cohort, but it is, I just want to emphasize that this is the gene amplification or copy number gain greater or equal to 6 per our standard criteria. And we are looking at germline as well when we're looking at genomic traits, and this is also in collaboration with CMAC. And then the transcriptomics, we're looking for signatures. We're looking for the pattern of immune modulatory genes, and there are many different signatures that are out there. And then we know there are signatures that are out there. There are now CLIA certified, Medicare reimbursable. So the field is really moving fast, and we are focusing on Wnt beta-catenin, chemokine gene pathway, HLA transcriptomic expression patterns, which already have shown to be linked to differential immunotherapy response. And it is currently being applied to clinical trial in a prospective manner. And this is one of the gene expression profile. And this is really the simplest one, which is just incorporating the PDL1, CXCL9, and interferon gamma, just a 3-gene combination, was able to predict that the high effector T cell signature was able to be linked with a better progression-free survival, as you can see in the graph, which uses atezolizumab as a frontline therapy. And there are many other genes of interest, including epigenetic regulators. And then we move on to immune landscape. Yes, we are going to look at the immune cell infiltration patterns. Also interested in looking at the MDSCs, Tregs, B cells, macrophages. And then we are using the PD-L1 DACO 28-8 monoclonal antibody. This was the one that were used for nivolumab trials by BMS. And we are also scanning all these images, and we do have plans to use digital pathology AI to analyze the image of these immune cell distributions. And PD-L1 is reported to be linked with overall survival benefit in nivo-ipi trials, renal cell and melanoma experience. As you can see in Checkmate 214, down table shows that PD-L1 positive group is the one that has actually benefited from nivo-ipi in renal experience. So very interesting interplay happening there. And then the proteomics, what's interesting is when we talk about protein, it's not really the proteins that's expressed in tumor or nearby tumor microenvironment, but it is also the host proteins produced by the host liver, acute phase reactants. And the mass spec is able to analyze the pattern of these spikes using the AI algorithm. So using that MALDI-based proteomic signature, we could actually identify the ones who have good survival outcome treated with nivo-ipi in this presentation. So serum host serum proteins and germline mutations are really the host factors. And then we do know that host neutrophil, even like if you look at the CBC and you look at the neutrophil count and the lymphocyte ratio, we've all seen many papers that yes, those are predictive of benefit from immunotherapy and even BMI. So there are host factors, and we are learning more about the gut microbiome. Although we don't have that piece in our translational medicine, but it is just the just telling us these also tell us that the host factor is probably as important as the tumor factor when determining response from immunotherapy. So how do we integrate all the data we're going to acquire from these TM collaboration? I guess the keyword is orthogonality, that these a lot of the predictors are independent of each other, and we really need to work together to form an algorithm to understand and weigh different variables to form a robust predictive algorithm. And understanding some are just bottleneck. Like I said, you could have TMB high, MSI high, but you could have a bottleneck, the JAK1 mutation, you have it, loss of function, you won't have any response. That's it. So I guess that there's got to be an algorithm to weigh that factor in a differential manner. And these are the papers that have consistently shown that T cell inflammation expression profile, there's a PD-L1, there is a tumor mutation burden, nothing correlates with each other. They are independent of each other. So I think there, some of the clinical trials support that finding. And in LungMap study, SWOG committee, we've seen that nivo-ipi versus nivo alone in subarm of the LungMap study. This trial was this arm was closed, showing no clinical benefit. However, the exploratory subset analysis showed potential overall survival benefit of nivo-ipi over nivo in patients with high TMB and low PD-L1. So this, I feel like, tells us that yes, finding the right group that will benefit from the combination approach is going to be the key. not everyone's gonna benefit, and it's just it's I guess the failure or the success of the trial will be defined by how we define the target population. And that's been the long experience as reported by Dr. Rezanova. And this is the final checkmate 227 survival that showed a benefit regardless of the biomarkers at this point. This was so the bottleneck I said you can have all the good interesting immune the pro-immune inflammatory findings on the left part, but then you can still have defective internal machinery if you have a genomic finding that can be a bottleneck for any immune activation. So I want to emphasize that yes, we want to look at the tumor heterogeneity, and this is talking about the importance of understanding the clonal structure. Is there a lot of subclones? Then you probably will not have durable response. So understanding the heterogeneity, not only the tumor TMB, those are two different concepts, and understanding those at the same time will be really important. So I think what's really good is that we have the pretreatment biopsy archived and the 3 different time points, baseline, first imaging, and at progression. So we're sequencing it, also looking at different change in cytokines, proteins, and from the tissue, we are imaging everything, immune cells to different proteins and sequencing RNA, DNA. So these are the basic TM projects, which will mostly be done by CMEC, as you can see. We have 2 commercial partners. Biodesix will be looking at the serum proteomic signature. Circulogene will be looking at the ctDNA as well as MSI high in the blood. And apart from the industry collaboration, we first started with Jack's partner and NCI, the same lab that NCI Match was using, but then we now partnered with CMAC, chosen as their pilot trial, and we're honored to be the first selected immunotherapy trial for them. And MD Anderson is our main lab doing the CMAC project. So as you can see here, cytokine assay, CyTOF, multiplex IF, multiplex IHC, RNA-seq, and soluble serum biomarker analysis will be done at Mount Sinai. And we just recently got approval for TCR clonality assay. And then obviously whole exome sequencing will also be done. So the serum assay is termed Olink, and this is the one that will go to Mount Sinai. And CyTOF is the one that will go to Stanford, but all the other pieces will stay at MD Anderson. So that's the CMAC site number 1, but they have 4 different sites, and all the data will actually be funneled into CIDC, which is headquartered at Dana-Farber Cancer Institute. So we're very excited. We have our first cohort being sequenced and analyzed as we speak. So we've prioritized group into 5 different categories, but I can tell you the first 3 groups are 3 neuroendocrine. We started with this is the very first group we reported the exciting response. And then we have separate pancreatic group, and then we have made a separate high-grade neuroendocrine group. So there are 3 different neuroendocrine groups. So we're going to look at that and run that group cohort as a pilot project and troubleshoot later. But then we plan to analyze samples coming from closed cohorts that have at least 2 patients with objective responses, and then move on to at least 1 patient with objective responses. So I'd like to thank everyone for this communal effort. Our committee chair, Dr. Kurjrak, and my partner, Dr. Patel, and everyone, especially from CMAC, who has been really on top of things, getting things to approval. There's so many moving pieces, but we've learned so much from this collaboration, and it's still ongoing. We're just beginning to analyze the samples as we speak. So we remain very excited and very thankful for all your collaboration and support. Thank you.
Thank you. Do we have any questions? I'm going to have some questions, but first of all, what you've done is like incredible. And hopefully the data will really help us better understand what is going on in these patients. So I think the potential is just unbelievable. I'm going to quibble with you on your TMB. So the first point I want to make is that we actually think that TMB is the single most important biomarker maybe ever discovered. And not to sound chagrined, but you didn't present any of our publications on that. So we've published several papers on that, and I wish there was a screen share, but it shows that our papers show that pan cancer, TMB is actually very strongly correlated with PFS and overall survival. And in addition, as a clinician, some of these patients have the best responses that I've ever seen in my life. But what I do want to go back is to that letter in New England Journal of Medicine. And if you can put that on again, I want to point out something. That recent letter that said that TMB may not be related depending on, because we did analysis of that letter for, I think it was Genome Web. And I did want to point something out about it because I don't think the data in the letter is as clear cut as people are making it to be.
Are you talking about Tim Chen's group?
Yeah, the forest plot.
The Annals of Oncology paper, right?
No, it's the New England Journal of Medicine, and it's the forest plot. If you can put that back on.
Across 47, yeah, I see.
Let me just point out something that I think is, if you, yeah.
And this one?
Not that one, the forest plot, which would be, I think it's the next one, the next slide.
This one's the letter. Nature.
Yeah, this is great.
Yeah.
So what I wanted to point out, if you look at the top of the forest plot, it's actually for all patients. Oh, that's just the bladder. If you look at the top, right at the top, that skinny little thing that looked at all samples, it's definitely significant. And if you look at some of the individual tumors, that's where you lose the significance. But if you want to actually, it's not on here, but if you look into the data, what you will find is that first of all, if you just look at it visually, they all move to the left, which suggests that they're moving in the right direction for significance per type of cancer. And then if you go back into the paper, and you don't show it here, but one of the problems is that in some of these groups, the number of patients with high TMB is extraordinarily small. Like we're talking about 10 patients with high TMB. So even though the forest plot moves to the left, the confidence intervals are extremely wide. And I think they're extremely wide because the number of patients with high TMB are actually so small. And so you're welcome to go back. We actually did this analysis for Genome Web, and you're welcome to go back into the paper. But I did want to make that point that overall they do show the high TMB as significant, that all the forest plots lean to the left, and that if you go into the details, the number of patients is exceedingly small in some of these groups, in the subset of patients with high TMB. So that's my quibble with that New England Journal of Medicine letter, and maybe why it ended up being a letter. But I'll stop there. But overall, I love this effort. TMB is not going to be a single marker. It's just that we really, our data show it's such a strong marker, at least for anti-PD-1, PD-L1 by itself.
Yes, thank you for the great comment. Yes, I do think TMB high is a pan-tumor marker. And I kind of envision this as something similar to MSI high. Like if you're above certain threshold, it is truly strong biomarker, unless you get the bottleneck that I discussed. But then, let's say TMB low, if you belong to that category, like if you're like already like low group, having low or low high, that kind of differential difference does not impact patient outcome on immunotherapy. I think I've looked at, for instance, like if you have MSI stable, does it matter if your MSI score, the number defined by, I used the Mantis score, but how does that impact survival or any immune infiltration difference? The answer was no. But I'm thinking, yes, very TMB, if it's really high, it's going to drive the graph to the left side and thereby being a strong predictor. But if you belong to the low group, then the small difference between different tumors and TMB level is not going to affect in a continuous variable manner. So
what I want to say is we did publish in Cancer Immunology Research probably about 2 years ago, where we looked at microsatellite stable patients with TMB high, and clearly they respond just as if they're microsatellite unstable. It makes no difference at all. Now, where the other thing that we found was that there was a linear correlation between TMB and response. And I think what one could quibble about is, should the cutoff be a 10? Like, what about the patients between 10 and 20? Our personal experience is that there's responders there. There's just not a lot of responders there. That the responders in the TMB 10 to 20 may be about 15%. And then when you start to get over 20, now you start to get much higher response rates, actually north of 40%. And then the question is, should patients with a 15% response rate have access to the drug? And my own concern is, because we've definitely seen responses. We just reviewed a patient with actually pancreatic cancer who had a TMB 11 and 33 months ongoing response. But I think the thing is what I worry about is if the threshold goes higher, the only people what'll happen is that people that have the means will still get the drug, and people that don't have the means will no longer get the drug with the 15% response rate. And certainly we do approve a lot of drugs with 15%, actually a lot lower, like Lonsurf, my favorite drug, 1.5% response rate. And it's true. Anyway, so there's some questions here. Or other questions. Anybody want to ask any questions? Okay, so in that light, I think we need to move on. But Dr. Chae, again, I think this is phenomenal. This will form a TCGA of rare cancers, and the ability to really integrate multiple markers into the Nivo-IPI paradigm. So I know you've put an enormous amount of work into this, and I think we all appreciate it. So the next speaker is Dr. Sandeep Patel, who is the PI of the DART